Hello everyone, welcome to the channel Learner's Wisdom. I'm here with a new topic, that is proteins. So which are basically biomolecules or macromolecules, specifically because on the basis of their larger size, and these are constituent of a number of things in the living organisms, like for example egg albumin, hair keratin, hemoglobin. Almost 99% of the enzymes are proteins, so you name it, and all the things are, a number of things are made up of the proteins: the cytoskeleton, the digestive enzymes, the enzymes of any metabolic pathway. So all these things are made up of proteins. So where from these proteins are coming? So it is actually when DNA, that is the hereditary material for all the organisms, leaving aside only viruses. So when DNA is transcribed, then it forms this mRNA, and when mRNA is translated, then every triplet code, it codes for an amino acid, and when the amino acid, then they make peptide bonds with each other, then they generate the peptide chains. And the longer peptide chains, when they attain a specific structure, then these are called as your proteins. So that is the basis of the life in the living organisms. So before we move on to the how these proteins are being made, how the peptide bonds are generated, they are formed. So let's discuss very briefly about the amino acid, that is D and L form of the amino acid, or the isomers of amino acids on the basis of the position of the amino group. So that is NH2. So if NH2 is on the right hand side in an amino acid, then it is called as your D-alanine, and if it is on the left hand side, then it is called an L-alanine. Where, and in this there is a significance of telling you this, because the nature has selected L form of the amino acids in the majority of the protein formation. D form is very less used. So now this is the categorization of the amino acids. Overall, there are approximately 300 amino acids that have been so far found in the living organism as a constituent of the proteins, but majority is these 20 amino acids which constitute the proteins. So that is why every book it suggests these 20 amino acids. So this is the on the basis of a specific categorization, that is positively, negatively charged, polar, uncharged, polar, charged groups. Then we have hydrophobic amino acids, some aromatic amino acids. So it depends what the criteria of categorization is. Accordingly, the amino acids can be placed in different categories. Now coming to the one letter code, this I have given because these are the standard nomenclature, standard one letter code for the amino acids, which you may find everywhere in any standard databases. Now coming to the peptide bond formation. So when, as I said, when this mRNA is translated, then during this process, when there is peptide bond formation between the two amino acids. So that is formed when between this carboxylic group, that is COH group of one amino acid, to the amino group of the next amino acid, thereby after releasing of the H2O group. So that means it's a condensation reaction, and that is why ultimately a CONH bond, that is the peptide bond, is being formed. So this is the small peptide chain that is showing CONH repetition of the CONH bonds in the peptide chain. So that is how ultimately a protein chain is being formed. So that is all for today. Thank you. I'll come up with the different structures, different level of structure of the proteins, that is primary, secondary, tertiary and quaternary. Thank you all.